let's start to try to be serious. Okay, I share my screen. Here, host disable participant screen sharing. Elodie, give me the power.
Yes, wait a minute. So I need to make you able to.
To share my screen.
Yeah, but I don't know how I can do this without losing my rights. It's a crazy stuff. Permettre d'enregistrer? No, I don't think it's this.
No, I don't know.
Okay, so you are gonna be animator, and you will have to give me rights again if I have something to,
Okay,
but it's
you
okay.
will tell me how to do it, but
But
okay.
maybe I don't need any to show anything.
Alors, meet you challenge one, share. Okay. Uh, so you have been a lot on the Discord to ask me some question about how to play with my 3D model to make 3D image from IC. I will speak about that in 5 or 10 minutes, but just before, I want to remind some basics and some help on how you can visualize an IC contact map clearly, what you have to do, what is some part, and just some biological reminder. Just as a start, because it's the morning and I like to be slow. First thing, what is important, because you are French people, you all know this blog, which is the best, of course, because you might probably already see that I give you a lot of tutorial writing last few years on that blog. So just go on it, look at my account, there is everything on it, like what's a compartment, how to download data for NCBI. This one is particularly made for trainee master students, so take an eye on it. And now we just go back on the Meet You challenge. Just a global reminder, there is two teams. One have to look a pattern which is sub-compartment, which is a phase separation between heterochromatin and achromatin in the nucleus. It's a pattern that we see at around 1 megabase resolution, or maybe less. It's a huge pattern, so we have to check huge window. But if you zoom a little bit, and you can see some structures which are size of 100 kb, it's the loops, the domain, just a huge mass of chromatin. And the goal of this teach is to understand the genome scale organization. For this, I highly encourage you to look at a YouTube video about loop extrusion. It's mostly for that team, but it's kind of interesting. It's about how loop and TAS are made. For this, they are made by two proteins. You just go on YouTube and you tap loop extrusion, and you will see beautiful movies with some screen here. And all is about coenzyme and CTCF. Coenzyme, it's a protein which form a ring. And this ring, DNA will fall into it and get loaded into it. And after this, it will try to find some CTCF pattern, which is just a pattern on the DNA where CTCF are bind. It is a directional pattern, so you have CTCF in the whole genome, and when two CTCF sites in opposite strand go to a coenzyme ring, it forms a loop. If there is multiple CTCF ring, it's not passed one through another, it just collide and it makes a huge mess, and these mess are kind of tad, in fact. But in fact, you can see that the comprehension about why CTCF and coenzyme did they make compartment or only loop, and this question about is these two patterns are linked or not, it's still confusing in the literature actually. Some people said loops and compartments, it's just the same construction at different scale, and some people said it's just completely different pattern and they did not have any link. Here it's one figure that I really like from a paper where they just tried to figure out what happened without CTCF because they made some knockout on this protein. And we see that compartmentalization, so phase separation between heterochromatin and chromatin, are still preserved. You can remove CTCF, so you can remove chromatin link, just loop and extrusion and all of that pattern. They will still have compartmentalization. That's why they think that there is completely different pattern. But some people said just, okay, you have one loop, you have one domain, you can say this is heterochromatin or chromatin and makes the phase separation along the whole genome, like in Bonnef et al., which is a seriously good paper in the reference. So there is some opposition in the literature about that question. That's why we try to make you sensitive about that kind of question, because this pattern describes architecture in the nucleus. And you have to understand that you have to detect them carefully as bioinformatics student. And after the link, it's still some work ongoing, and we don't know every question. Maybe you will ask me some question on the Discord about epigenetics, and I did not have the answer. And I've made 3 years of PhD about IC, so sometime we know, sometime no. Now I will speak a little bit more about the operation that you have to make on your matrix. One question that I have twice in the Discord actually is about the resolution. The resolution on the IC matrix, I give you some data at 25 kb and 100 kb. 25 kb, that means all of the genome are of block of 25 kb. So I just take my chromosome, divide them in block of 25 kb. Just now, as an example, I will just go on my GeneRef. Chrome size. As an example, basically, the chromosome 1 of human. Alors, attends. Comment je repasse dans le chat?
Le chat, il est en bas normalement, non?
Attends, il faut peut-être que j'aille dans la vidéo. Alors, mort, chat, trouvé. Alors, I give you on the chat the size of the first chromosome of human. So, in order to know the size of this chromosome at 25 kb, you just have to make this size divided by 25. So it will give you the number of bin you have in your dataset for this chromosome. So here, basically, it's just, for example, at 100 kb, it will be 3493 bin à 100 kb. Parce que du coup, tu divises, là, il fait 249 millions de paires de bases. Yeah. This size is the total size of the chromosome 1 in base pair for human. So if you divide it by your resolution and you make a round of this value, you will have your exact number of bin of your chromosome at this resolution. It's just a basic division. But it's important to remind, just because it's how we see the genome at this scale, because we cannot go at 1 kilobase or 2 kilobase, because you will need a lot of signal, you will need to sequence a lot, and it's really expensive. So you cannot have that kind of information of quality. So you make something more global, but you already see a lot of pattern. For compartmentalization, it's enough to see it at 25 kb or 15 kb or 100 kb. It's okay. You will see almost the same thing with little difference, which is a challenge for the compartment team. So it's just some way to think about, okay, we see the genome, we have this size. This is how we calculate it. It will help notably for if you want to make 3D, because I use that kind of tricks a lot, in fact. So when you have that kind of operation in mind, you will know that your map is at a resolution of 25 kb by an example, but you can change the resolution to a bigger one. So that means you can go to 25 kb, no, from 25 kb to 100 kb. So for this, you will just have to make the sum on all region of 100 kb along the genome. Remember that the matrix is symmetric. So here, for example, you have a small matrix of 25 kb on left with the red square, the blue square, the green, and the yellow. And if you aggregate it at 100 kb, that means you need... attends, c'est faux en plus là. Oui, c'est faux. Là, là c'est pas de 25 à 100, ce serait plus de... Here it will be more 15 to 100 kb. I'm sorry, just that here you have two blocks. So here, by example, you just aggregate twice. So if at the beginning it was 25 kb, after this procedure it will be 15 kb. I will have to make a matrix with 16 square to make the operation from 25 kb from 100 kb. Is that okay for everyone?
But
I maybe not have been clear at all. I just...
For 100, it will give one cell, right? Just one cell.
Yeah. C'est... Tu divises la taille par 4, basiquement. Parce que you just go from something which is 25. So to make 25 to 100, it's just 4 blocks of 25 kb.
So meaning that, yeah.
I have a question. What if the last bin it's not enough 25 kb?
You will make one bin more. That's why every time you compute the resolution, you compute it with your chromosome size, with the good resolution, and you will see that the last bin, it will be just...
I put a 0.
What?
I fill the every value in 0.
Yes, the last bin will have less contact maybe, but it's resolution effect. You have something homogeneous along the genome except the last region. So if you want to look carefully on it, maybe you will have to choose a resolution that is better.
I choose it's not 0.
What?
So the value for the last bin, the last bin it's not enough 25 kb. So I made the value it's 0 or 1 or nothing.
0?
But if you want to complete, yeah, you will consider the rest is 0. But what Leopold was saying is that it means that this region will be at 100, it will be poorly covered, it will be not realistic because you put some zeros. So if you want to take a look at this region carefully, then you need to stay at 25 probably. That's just the border effect, right? The border effect will make the regions at the border less reliable when you do this transformation to a lower resolution, right?
Alors, just here for an example. So it gives you 25 bin with one. That means the last bin contain only one block of 25 kb. The matrix size will be 9971. Okay, I made a comment just to formalize it a little bit.
That's not my question. My question is for the last bin, the value for the last bin for my case, I made 0. I just put 0. Je mets 0 pour le dernier bin. Voilà, celui-là, c'est
Non, tu mets pas. Tu mets pas des zéros, c'est juste qu'à l'agrégation, cette région-là, elle va être, elle va contenir moins de signal, c'est tout.
Signal, c'est tout, mais le dernier, c'est pas suffisant le 25 kb. D'accord.
Ah, if it's not enough, yeah, it will. Okay. I will make a paint. A new paint.
Yeah, maybe you can draw what you mean with the question on the sheet.
Il a compris. Il a compris, je pense.
Yeah, now attends. New share. Desktop. Okay. I think now I have it. So you have your genome like this. You divide it by block of 25 kb. And his question is because, okay, ça va pas être des blocs de, it will not be. What did I do with, with this part? And especially this part, yes?
Yes.
But you will just have less signal in your last bin.
Yes, the last bin is not enough.
Yeah, it's not enough, but it still have the
still
part.
Well, yes, still have some data.
Yeah.
So the last, I just made a 0 because it's.
You not put 0 on it. You just put the signal that you have. You just create a matrix.
Okay, I see.
Of
So
the size
I don't care.
need,
I don't care
with the information that you
size.
have. So you will not put 0 on it. You will just say, okay, I have that amount of signal on my bin.
But yeah, it's equivalent to having zeros in the part that is not covered.
Yeah.
Of course. But this part doesn't exist in that case, so.
When you create your matrix, you have reads. Reads are of 200 base pair. So you just count where they are and you put the number of time you see it every time on each bin. So if I make a draw, it's will something like you put some read here, you put some read here, and you just count for each region how many reads you have. So you will just put your numbers that you have, and yes, okay, it's not complete, but it's the last bin, so it's a compromise. Last bin are not always perfect.
Okay.
Otherwise, you crop.
So the matrix, it's not the same. C'est pas la même taille pour la matrice.
The size of each block will be the same, but the amount of information that you will have on the last bin, okay, it will not remain with the same size of information, but it's a compromise of resolution.
Okay.
Otherwise, you can crop, no? You can ignore also this part if you think it's not reliable, I guess.
No. Okay.
I don't think it's really important, in fact, because there is always less signal in the extremity because it's telomere information. So at the end, it will be just removed. On maybe next step. Hop. Attends. Alors, zoom, new share. Alors, go back on the diapositive. Share. Yeah. No, I didn't have filtering here, but I will speak about filtering in 10 minutes, and maybe in filtering this step disappears. Yeah. So yes, when we will speak about filtering in 5 minutes, maybe it will help a little bit just because that kind of region are really low in signal, so you just remove them. Is that okay?
Okay.
Okay. So next step. Here, it's just some example about how I like to visualize an IC matrix. When you see some raw data, first thing that you have to make is a color scale gradient, which highlights more the diagonal, because there is more signal at close contact than long distance interaction. Here it's an example about Arabidopsis thaliana, just a plant, because I have some data in plants with chromosome 1, which are here, the chromosome 2 are here. Did you see my my mouse or not?
Yes.
Okay. Here it's chromosome 3, here it's chromosome 4, and here it's chromosome 5. Here the resolution is at 100 kb. And what you see here is the chromosome 2 at 10 kb resolution. So that means that all you see here is just the part exactly here. On this species, each centromere interact with each other. That's why you see here the centromere of the chromosome 1 interact with chromosome 2, with chromosome 3, 4, and 5. And that's made the pattern here. When you see that kind of matrix, look at it at log 10 scale. It's really important because you have logarithmic scale on your information on your matrix. So if you just see it as an image and said, okay, I put the image, the scalar image, you will see nothing. If you put it in log 10 scale, you will start to see something like this. This is really important. Okay, that's example. And one thing that I think it's important to say is, what is a good color scale for visualization? One thing that I advise you is to make a color scale from one color to another. Don't make something with blue from white from red, because here, by example, you will have a transition on a value which will be completely random, and you don't want this. You want something that is really continuous to your information. So from all of these color scales that I made, I prefer this 4-1 than this 2-1, which for me are kind of tricky because you can change the axis and you start to white, you finish to white. So what's the point? This information is good for TAD because you will see mostly raw map. But if you want to like compartment after all of the pipeline of normalization, you have something with negative value and positive value, which are centered from 0. So for this, you are 15. So basically, there is at least 1 or 2 Daltonian people from you. And so for the Daltonian convenient color scale with correlation matrix and something which go from one color to another, I highly encourage to go to red to blue, because it's the best way for this map to be seen easily for everyone. Make it centered. That means the white is at 0. It's really important because in other case you will confuse your pattern. Remove the null and the infinite information. In other case, it will be just a huge amount of blue, and like here, basically it's the chromosome 1 of human. And in the centromeric region, I have absolutely no signal. So when you make a correlation on 0 information, you will have none on your matrix. So I just replace them by 0. And one other thing that is important is to plot your matrix as a square. It's just because you want something homogeneous from the axis X or the Y in order to see the same information. If you plot it as a huge rectangle, it will not clearly easy for you to make the pattern and see, okay, did the information that I make are good or not? If all of these basics are okay, I think I can go a little bit on the code. Some of you been on my GitLab and look a code that I made for a paper that has been not published yet, but one day it will be. So what I propose you today is to just read a little bit this code together in order to understand the basics. So I will just go to the code. Did you see it? It's okay?
Yes. It's okay.
I have bad news. It has been right in order to be almost similar line by line to a MATLAB code. So it's not a Python code. It's something that is between Python and MATLAB, just in the way I write it.
Excuse me, can you introduce the data file? Because I didn't understand the file load observed.
What?
The file load observed.
Attends, I did not understand what you said.
Je n'ai pas compris le fichier low observed.
Ok. Alors, your data, what you have. Alors, I will just compare with what you have. Alors, GitHub, give me 5 seconds. Alors, meet you, data for student. I see. Maintenant. Alors, okay. Why there is row observed on the name? It's because if you look on the data that you have, the names are kind of similar. It came from the paper from Rao et al. And sometimes they made several normalization on the matrix, and they give the row observed data, the row expected data, and the data after normalization. So I just give you the pure row matrix in order to give you the opportunity to make the normalization step by yourself. That's why in the name there is row observed. If you want the row expected, it's after some operation when you make compartment organization, by example. When you divide each point on the diagonal by the mean of the value, it's the expected value of your matrix because it means that it's homogeneous. I think. I don't remember why they make expected and all of this. I just download the pure row matrix. So
the file just exists 3 columns. So
yeah.
First column and the second column and the third column. What does that mean?
For this, we have to go back on the first meet you teach. The first column, it's the coordinate in base pair.
Okay. So the second is elect.
Of the first part of the interaction. The second column is the second part of the interaction. And the last column is the number of time you see the first region with the second region in interaction. Your IC matrix describes the count of each time you see each interaction of the first column with the second column. So basically, I will show you a simple code. I have something.
So that's its image. See an image?
Yes, yes. It's the coordinates of your cell and then the value in the cell. You can see it.
Basically, if you want to make a matrix with it on the simplest way, here an example.
Okay.
You see it? You have your file name. You have the size of your IC matrix.
Okay.
You have to make so it's just chromosome size divided by resolution and the resolution of your block. Okay.
Okay.
So I think it's a simple Python code, so you will easily read it. First, you open your file, you read each line, you create a matrix which is symmetrical with your IC size based on your resolution.
Why should the divise résolution?
What?
Pourquoi on divise les résolutions?
IC size. The IC size, it's your chromosome size divided by resolution. You want at this line?
Yes.
Give me time.
Okay.
It's the next operation. So first, I will maybe open a dataset. Alors, teach. Meet you. Data for student. Alors, IC. GM68, intrachromosomal. Alors, aide, CHR5, hop là. Basically, you have coordinates like this. So coordinates are in base pair. So you want to convert your coordinate in base pair into matrix coordinate at the good resolution.
Oh, it's a normalization, okay.
Because you see your genome from a resolution point of view. So that means you are seeing it with a matrix which are of a specific size, and you want to know for each base pair you have where it go on the matrix at this resolution. So basically, if I go back, alors, hop là, on my whiteboard. Ah, attends. Ah, I have whiteboard here, but no. New share. Ah, dégage, c'est nul. Alors, share. I will go back on paint, just like this. So basically, just said that we are at a resolution of 100 KB, just to make it simple, okay? Alors, first thing that you have to know is that, so your first block of 100 KB, so it's start at 0, and after, here, it's 100 KB. After it's 200 KB, and here it's 300 KB, and you continue bin by bin, so by block of 100 KB. So if you want the index on a matrix which are at a resolution of 100 KB, basically, here it's a 0.0, here it's a 0.1, here it's a 0.2, here it's a 0.3. Do you understand that?
I understand it. I understand
so it. Because you go from base pair to index on your matrix, you just divide each
resolution
column by the resolution.
Okay,
and
thanks.
All the information on the data are always already at the good resolution, so you don't have to make truncature or to make round. That's why the code that I show you now, new share, apply, IC toolbox, share, are good. It's just something simple. I made a lot of cast because I was just lazy and write it fast, but it was just to make you a simple example. And so after this, you just add on your matrix with your begin coordinate and your end coordinate, so your first column and your second column, the information that you have on the third. It's symmetrical, so you put it on the 2D direction, and after you have your matrix. Okay?
Okay.
Now, if I go back to the code from Shrek 3D, it will make basically the same thing, but with more mass. Because it has been written by a physicist who love maths. But basically, all this line, it makes the same operation, but with sparse information in order to first, it tried to build the array at base pair resolution. So you give him a matrix at 5 KB resolution, and it makes it at base pair resolution. So it will extend the matrix in order to change resolution from the point that you want. R here, this parameter, I have to note it, it's your resolution. It's your size of block. Here it's some parameter for our paper just to play with epigenetics, the same that I give you. And it's the same file name. This value, it's kind of tricky. I advise you to didn't change it at all if you want to play with that pipeline. We have parameter it for human matrix, so it's okay. I will explain you just soon why this value is really important. It's about theoretical physics and dimension reduction, so please don't play with it. So basically, first, you build your matrix. After all of this line, you don't necessarily need to play with it because it's just a way to play with the epigenetic dataset. And now, if you understand that here, all this line is just the build, basically you build your dataset at the specific resolution that you want. So if you want to play with it, just change it by a simple loader, and at 100 KB, it will be completely enough. Just remove the epigenetic information. You don't need it. Or at least if you are in compartment team, you can play with it a little bit. But basically here it's just load the annotation about epigenetics and count how many times you see each epigenetics information on your genome at base pair resolution, which are converted then to the good resolution. And now we will go to what is really important for you. First thing, I will go back, new share to this one. First thing that you have to know, on your matrix, the signal is not homogeneous because each line and each row didn't have the same number of information after the experiment. It came from a lot of different biases. For example, it can be the GC content, the mappability of your matrix, because you have a lot of repeat region somewhere and somewhere not. And this can mean huge amount of difference on all your line of your matrix. So your signal have some region with completely nothing. By example, if I draw it just by hand, the histogram of the sum of your line will look about something like this. So that means you have two information that can be tricky. You will have some region with really low contact and some region with really a huge quantity of contact. Attends, je vais faire ça un peu propre en mettant un 0 par là. Et l'autre. So this two part in red, you don't want it because if you have to less signal, it's not significatively enough, so it just gives you nothing. So you want to remove it. So the best way is just because you have a pretty Gaussian model. I go back on my code. I'm sorry, it's the first time I teach online. Or the second with you, but just okay. So you just compute the mean and STD. It's just le theta, donc la variance, the variance of your data of your matrix. Here it's just a ratio about how many parts you remove, but you basically just want to remove the less 5% and the top 95% bin of your Gaussian distribution. And here it's just some line to filter your matrix and remove this broken line. You do the same with the color. And after we can go to the real part which make the 3D. Are you okay with the filtration? Did I have been clear? There is only two people who speak.
Okay.
Is that clear for everyone else?
Yes.
Okay. So now, first thing that you want to change is just that you don't have the same number of information from each line and each row, and you still have a Gaussian information. Okay. Attends, je vais retourner sur mon code. So first thing that you make is sequential component normalization. I give you the code somewhere. I think you see it. SCN. Do you see the code of SCN?
Mm-hmm.
So what you want to do is to make homogeneous information from each row and each line. It's better to visualize it when you show an heat map. Make an SCN before. It will help you just to see it clearly and to see all the interaction correctly. If you have some loop that you want to see, it will just highlight it and it will be better to see. So the code is just you make the matrix have the same number of contacts for each row and each line. So it's an iterative correction, which makes in sort that all the sum of your line will have the value of 1. Sorry for this. Alors, so to do that, you just have to take your line and you divide it by the maximum sum of your matrix at each row and each column. Max iter is the number of time you make it. Basically, here I put 10 because most of the time, 10 iteration about divide each line by its sum, divide each row, each column by its sum. It's enough for 10. There is the matrix start to be stable at 8 and it's finished to be completely homogeneous at 12, I think, but from 8 to 12, it didn't change anything. So it's the first operation. After that, this where alpha came, you will try to convert your contact map to a distance map. Because what you have is a matrix where you count each time a agent has been seen in contact with another. So it's not a distance matrix, it's a contact that you've seen during your biological pipeline. And you want to convert it as a distance matrix to have the distance with all of the points. Because if you have all the distance from each region, you can say, okay, I have a distance matrix, so I can make a 3D model with it. This is the game. So for this, we will convert our contact matrix to a distance one. For that, the alpha parameter will just make a ratio about how much we take about the information that we already have in the matrix. Remember that one thing that I've said last teach is just when you make IC, it's not single cell IC. It's made on a cellular population. That means all the structures that you see came from maybe completely different genome organization. So your IC matrix did not reflect one cell, did not reflect exactly a cell type. It reflects the mean contact of all cells, the cell that you have sequenced. We hope that they are homogeneous and at the same step, but sometimes there is maybe some different conformation on your DNA. And so in order to not take all of this information which can create conflict with them, we have to modulate it in order to make something that will be pretty. That's why this alpha parameter came. If you want to have more information about it, I can give you the reference. It has been made by my PhD advisor, which is Julien Mosiconacci, just right here. So it's polymer physics, in fact. Now I need some matrix to explain you. I need an extra link. Alors, l'équipe. I will change from the code to your favorite blog and go to alors, transformer une carte de contact. Alors, new share. Hop là. So basically, what did I say? First, you make your sequential component normalization. You put a neat map. Here it's not with good color scale. I was young, sorry. And after, you convert your matrix of contact to a matrix of distance. It will look like this. So now you don't have a contact matrix, you have a distance one. And you see that from the scale, there is some region where there is distance from 0 and some region where there is at least no information. All the regions that are in yellow here, it's just contacts that we don't have. Your matrix, your IC matrix is really sparse, in fact. There is a lot of 0. And what we want to make is to reduce the dimension of this matrix to 3 dimensions in order to see it in 3D. So for this, we will make shortest path algorithm in order to compute and have some information on each the points that are in yellow. So basically, it's just up, new share code. New share main. So we put fast Floyd in order to compute the short path on the matrix. Here it's just to be sure that the matrix is really symmetric. It's just some check operation. And when you have a complete distance matrix based on your IC map, you can make a dimensional reduction operation. So here it's with Salmon model, which is really good. And it gives you the X, Y, Z coordinate. And from here, it's just completely easy. You can just write it as a PDB. I made a function based on a code of Pierre Poulin from Paradidro, and it works completely. Do you want to see a 3D just as an example? It will take 2 minutes.
Yes, I like that.
Okay, alors, I will just go to my PDB data. I think I have some of them somewhere. Data, R, PDB, où est-ce que j'en ai mis? Alors, code, fonction note, biomol. Alors, quelle est la dernière? Parce que mes dernières ont dû marcher. Dernière modification. Parfait, j'en ai quelques-unes. OK. Ouvrir dans un terminal. So now I will just open a VMD, new share. Est-ce que je peux share mon VMD? Parfait. Desktop 1. Here it's my VMD. I just open it and I will load a matrix. So first thing that I make is just browse the molecule. I have some of them. Which are the last one? It's 3D color, machin, the set. Okay. I will load it. It will look like this. So now it's not really pretty. I will put orthographic perspective just because it gives you best point of view. I will change the display in order to make it clear. Background, white. So now it's not really pretty, but I can make something like this. Change the representation and go to tube. And here you have the 3D of one chromosome. I think it's the 10. I don't remember. But it looks correct. And what you see is really, it makes some goes back from one to another region. What you see on this 3D is the phase separation from heterochromatin to achromatin. This tool helped me a lot when I studied genomic compartments, because in fact, you will see that all compartment B are just on the same part of the chromosome. It's completely easy to add compartment on your 3D. You just have for this to go to write PDB and add it on the value of beta. So just one parameter. So for
B factors, right? The B factor column.
Oh yeah. I put my information on beta factor colon on the PDB, and after I just color my 3D structure by this beta value.
Exactly.
And you can color by compartment or add any information that you want, and just said, okay, with this specific value of beta, I can highlight something. So, as an example, okay, here I don't know what I have, but if I color by beta, I don't remember at all what did I want to do with this one. But you see that there is some region in red and some not. So it can be a good play. I think if I go back on the slide, I will show you something that is more beautiful. Up. Yes. Here it's a good example. It's an example that I made during my PhD, where you have here the compartment B in blue and the compartment A in red. And you see clearly the phase separation. But if I play a little bit and I create not 2 but 3 compartments, it will put another band on my chromatin. So you can completely make some pretty representation with that kind of matrix. If I put 5 states, you see really the gradient from red to green to purple to yellow to blue. So it's a game that you can make for the compartment challenge. You can play it with the epigenetics matrix that I give you. I give you some tips about how to do it. Yeah, but the challenge was about what is the best number of compartments, because here, as an example, you see that I can make 2, I can make 3, I can make 5, but in fact, the phase compartmentalization is just go from heterochromatin to a chromatin. But is there an optimal number of compartment computation? Rao et al. In the paper from this data set has been asked the question, and from this date, no one tried to go back on that question. Jobdecker, which made the IC, spoke about phase separation, but it just said A to B. They never increase this ratio. Why? Can we see something? It's an open question from the Me Too challenge, actually. I may have some answer, but I keep it in mind and just make you have fun about this. So for this, you have your epigenetic with active transcription factor, flanking, weak transcription, and answer, heterochromatin. So basically what you can do is just play with heterochromatin on some chromosome and check how much it changes along this gradient. You can play with some stuff like this. Here I give you an example of pipeline to solve this question. I think it will work. I'm almost sure. Maybe I already test it. But
so I have a question, Leopold. So for these epigenetic marks, what's the idea? You just take them blindly and you try them all, or can you already have some a priori about what they mean? Because they can be very diverse, no? They can they represent all types of information.
Yeah, I think the best way is to play with heterochromatin or active TSS, because what we know is just on the B compartment, it's completely heterochromatin. On the transition, there is some mark like polycomb. So the mark 13F40, we replace polycomb, it will put on the middle of your compartmentalization. And after you will see more and more active TSS, flanking active TSS, and strong transcription. All the green and the red mark go on the A compartment. All the heterochromatin go on the B compartment. So for me, what is the best way to compute the optimal number of compartmentalization is just to know when heterochromatin just didn't are present at all on your genome. So you just want to know when there is a break from heterochromatin to a chromatin. And did we have the same feature on each block? So you have to compute a number of blocks with really distinct features. It's a question of clustering, in fact. One way to solve it, maybe you can just do it like this. It's you have all your mark, you make hierarchical clustering on it, and you will see a number of clusters. This number of clusters maybe describes the optimal number of compartments. One another way, it will be more tricky because I have to make some remind. You have your IC matrix, you have converted to compartment matrix with all that I described somewhere here. Here it's your matrix after sequential component normalization. So for study compartment, you have to convert it to observed unexpected matrix. So that means divide each point by its mean diagonal, and after make a correlation of this matrix. And here you have a good correlation matrix. On this matrix, what you can make is order it by its first eigenvector. So you have your matrix, you compute your first eigenvector, and you order the first eigenvector, and you make the same order on your correlation matrix. And what you will see is you go completely from the B compartment to the A compartment. And here you have some region where the value of your eigenvector is really small. So your optimal number of compartmentalization will be somewhere here when you know where all this information on the blue are really separate from the information in red. So maybe it's 2, maybe it's 3, maybe it's 4, but it's just to check the information in the middle, how much it's separate from the block of B, which is really strong B, and the block of A, which is strong A. Did I have been clear? I think not.
Pardon, non, j'ai une petite question par rapport à ça.
Elodie, j'ai été bon ou pas?
But for me, it's okay, but what about the students? You cannot do some kind of... Il n'y a pas le pouce dans Zoom, non?
Ouais. Duncan, Quentin, is that been okay for you? Sukaina?
Est-ce que je peux proposer une question pour Challenge 1?
Pose-moi la question et je te dirai si tu peux la faire ou pas. Si ton équipe est d'accord.
Ok. Pour le premier Challenge 1, which part we should improve? Because I didn't see your code will have a top-down algorithm and arrowhead algorithm. I didn't see.
Okay.
So
okay.
Which exactly we want to do?
What I propose now is just we finish with compartment because we start with it.
Okay. Yes.
About study, and after we go on TAD. Is that okay for everyone?
That's okay.
Okay. So just some remind about compartment change. I give you my output, which is just a vector at the good resolution with positive or negative value for A or B compartment. And in -1 is the region that I filter by myself. So here to load it and as a reference and said, okay, that's what you have to make. It's just to give you an idea about how to do it. Reminder that when you make your compartment with your first eigenvalue, you have to check its sense. It can be on the wrong direction. So for this, I highly encourage you to play with gene density, or if you want, you have the epigenetics. If you check it by just active TSS, it will be completely okay. Okay. Your output, you already know it, but I just put back this slide just as a reminder. Did I said everything on compartment? Yeah.
In any case, those slides are on the GitHub as well.
All of these slides are on the GitHub. I just downloaded it from the GitHub back just to have a copy. And so you have everything that I show you today already. By the way, I add all the papers that you have download on the repository of download, just in case you not have access to it. So I think you have everything, almost. There is some challenge that I didn't describe actually on the compartment, just like you have your data at 25 KB and 100 KB. Is the compartment that you made at 2 or 3 are the same or not? Can you see the difference and where are the difference? Can you link them with the value of your eigenvalue, and are they on the noise or are they significantly different, and why? If you understand that question, it will be a really good point for you, because I made quite some time to just say, okay, what is the effect on the resolution on this operation? Another challenge is to play with interchromosomal contact and not intrachromosomal contact. If you play with intrachromosomal contact, it will be easy because you will just have one square matrix, like chromosome 1, you made the whole pipeline, and that's it. But if you want to play with interchromosomal contact, there is maybe less signal. Maybe it gives you another signal. Maybe you will have to adapt a little bit the pipeline. Are they the same? Did it describe the same information or not? It can be a challenge that you have that you can solve that are interesting. Ok, et là je fais vraiment le truc à l'envers, c'est pas grave. Ok, and I've tell everything. Ok, I think I tell everything on this part. So now we can go back on the third challenge. Okay, third challenge. Basically, the bricks that you have for that challenge are, from now you just have the loading operation, your second cell component normalization.
There is a small square on the right that we can see on your screen. At least I see it, and it hides a bit the slides.
Ça?
Yeah.
C'était le chat?
Okay. Okay.
Sorry.
That's better. It's okay.
Okay. So basically, for TAD, you have to start from the beginning because you just have your IC matrix. You show it at log 10 after second cell component normalization. It will give you something like this. All your TAD is just the square along the diagonal. You have to detect it from row. On the GitHub, I think there is arrowhead. There is here an example about a good way to compute it. I give you on the chat. This video shows you an algorithm about how detect TAD on your IC matrix. What you see on the matrix is really just a continuous a block of square along the diagonal. So that means between each square there is less signal than inside each square, and you want to detect this low signal region. Alors, est-ce que après les traitements faits avec votre code, la matrice devient log 10 ou non? Non, tu le fais toi-même. Tu fais dans l'ordre load, affiché en log 10. Hop là. Amira asks, after all the pipeline that I described, is the matrix are in log 10 or not? No. The operation for that team is just in that order. In order to see it clearly, you just load it. You make your second cell component normalization, and after you see it on log 10 scale in order to see it clearly. You can see it before SCN, but it will make some biases on the observation because each line are not completely the same, so it will not be the best for you. So keep in mind, before visualization, make second cell component normalization every time. And so what is interesting here, it's that kind of region where there is less signal on the data. You want to detect all of these peaks. They are informative. So for this, you can go on the YouTube link that I give you and start from the arrowhead pipeline, or start from the top-down pipeline that I described sometime, or you can start from what you want. Clearly, you can make some pipeline with some along each line and try to compute. I just give you some basics idea in order to solve the challenge easily. But if you want to start from the beginning and make something new, you are free. It's just the constraints that I give you. It's all the tags that you detect. I want you to be sure that they are good tags. So for this, the best way is just you compute your algorithm after sequential normalization, you optimize it, and you see it on the IC matrix if all the tags that you detect are good or not. So you just take by hand some time to annotate some coordinates and said, okay, this is my tag. In other words, we only need to detect the red blocks. Exactly. This is a way to say it completely. If you detect all the red blocks along your matrix, you detect the tag. But you have to be sure that you only detect the red blocks and not something that is not a red block. And I want you to be precise about what is the border of each red block that you detect. It looks simple like this, but trust me, you can spend a lot of hours trying to be the more precise that you want. Because you made it on the whole genome. Each genome has its own specificities. Sometimes there is some region with missing information. So like here, there is a huge gap. And so it will detect 2 tags and not 1. And all of this small sand on your algorithm will screw everything. And you can spend hours by manually annotate it. And you don't want it. You want to make something that is universal, just to have one match. I see data, load it, and said, okay, this is my tag. I can play with them. I can extract information. So this is a challenge about features detection, in fact.
Okay.
So, yeah, I highly encourage you to start from the work of top-down or arrowhead, because your reference for this challenge is the arrowhead from the paper. Top-down is a reference on the literature, and it's really simple to implement. It's just 15 lines. So 3 of you will just code in 2 weeks, and you will have the basics. But you have to choose. And one way to solve it, and that I proposed on the GitHub, is some reference about CTCF. It's why I speak about it today. It's because if you want to download by yourself some CTCF annotation, I can give you a link where you can easily download it in
what
easy
is
format.
CFC? What is CFC means?
CTCF is the protein that I speak at the beginning of the talk. Here, about this picture. CTCF is the protein which binds at each border of TAD, because it came on the loop extrusion principle. So if you try, by example, by compare your algorithm with CTCF chip enrichment, and where all CTCF protein bind onto your genome, and said, okay, I will try to detect only TAD which are enriched on CTCF, it can help you to improve your algorithm. It can be one way to solve this question. So this is a good way on the literature to just detect only TAD that you are sure. Because if your TAD are not trustful, what's the point? You detect something, but can you extract some biological information from something that you are not sure about? Can you make global observation, like, by example, size distribution of your TADs with noise on your data? Here it's an example for some species, but maybe here on the chicken, I have some small TADs of 100 kb. Are they short? Are they just a basis in the way that I just used top-down with basics parameter? That's the question. That's why I want you to make only good TADs. So basically, yes, just the red block on the matrix. Is there any question from now? I think I described most of the slide. I just didn't speak about the combined challenge. And the combined challenge is kind of simple. You detect your TAD border. The other team detects the compartment. Can you create back the phylogeny? So implement a hierarchical clustering or spectral clustering in order to establish the phylogeny from each cell type with these two features. So it just implements a clustering which works with binary information. And I think I described everything from the technical point of view. Did you have any question? It's a good timing. Now we are here for that.
Pour le code de CSM, pourquoi il est besoin de le 10 fois intégration?
J'ai rien compris.
Je tapais en un mot.
Ah, attends.
Okay. For the code, why we need to time integration?
Because it converges really fast and fast. In fact, after 4 cycle, your matrix will be almost homogeneous. You can play with it a little bit and just check your sum of your line at each operation, and you will see that after each step, it will be more and more a line which are really homogeneous.
Okay.
Just it converts really fast.
Okay.
So at 10 cycle, you are sure that your matrix is good.
Okay.
In fact, the original paper just said that you compute your number of cycles and you just check if the variation between 2 cycles are really small or not. I am a boring practical bioinformatics, so I said that after 10, for each species, I'm sure. So it's a lazy implementation of a more complex algorithm. And that will finish on YouTube, so. Yes, it's just a practical way to see it. Is there other question?
Oui. Est-ce qu'on m'entend? Oui, on
Oui,
m'entend.
bien sûr.
C'était pour revenir sur le eigenvector, au niveau du eigenvector.
Bien sûr.
À partir du moment où on recompartitionne les deux couleurs, donc le rouge et le bleu, enfin le bleu et le rouge, au milieu, si on crée plus de détection de compartiments, si on clusterise plus, on verrait une transition de couleur au milieu, mais est-ce que le fait est-ce que les valeurs qui sont aux extrémités des clusters écraseront pas les couleurs qui sont liées? Est-ce qu'on verra bien les couleurs qui seront la dissociation des couleurs? Oui. Est-ce que vous mettez l'intérieur?
Attends, j'ai des images pour te montrer si tu veux. Je dois avoir ça pas loin. HMM versus VP, on a H1, tananana. Alors varcomp, DKB. Non, c'est pas ça. Je vais trouver des images, mais il me semble que j'ai fait des visualisations pas trop laides de ça il n'y a pas longtemps. Parce que ma chef m'a posé la même question en fait. Ah, attends. Alors tiens, ça, tu vois, typiquement, c'est un exemple qui est bien, c'est sur le poulet, où là, regarde, tu as la matrice du chromosome 12, c'est de l'humain, et j'ai fait 6 compartiments.
Ouais.
Et là, tu vois bien que le compartiment le plus rouge, qui est là, du coup, qui est le chromatine, et le compartiment le plus bleu, qui est l'hétérochromatine, tu as beau augmenter le nombre de compartiments que tu crées, tu auras toujours ta carte de compartimentation qui est un gradient rouge-bleu parce que c'est ta séparation de phase sur ta carte de corrélation. Et ton nombre de compartiments, d'accord, tu vas en mettre plus, donc plus de couleurs, mais tu verras toujours bien les domaines que tu vas distinguer. C'est juste que ça va te mettre plus de distinction entre toutes les régions qui sont un peu en transition et qui ont des valeurs de corrélation qui sont un peu plus proches de 0.
Ok. D'accord.
Donc ça prouve que là, typiquement, tu vois bien, là, tu as le bloc, tu as le gros carré qui est là, qui est de l'hétérochromatine, et tu vas voir apparaître des régions qui sont un peu intermédiaires, et elles vont apparaître dans d'autres couleurs.
Ok.
Voilà.
Merci.
Je t'en prie. Maybe I have to translate it in English for the YouTube or not, Elodie?
Yes, maybe. Yeah, maybe you can summarize it.
Yeah, okay. The question was about when you increase your number of compartments and you show more compartments on your correlation matrix on your correlation map, did will affect or not the visualization? It will be confusing or not when you see it? And what I describe to the student in French, because sometimes we speak easily in French. I'm the first one in that case. It's just that here, for example, it's I made 6 compartments on that matrix with the 0, which are the heterochromatin, and the 4 in red, which are the 5 in red, which are the chromatin, and the strong chromatin. That your transitions are still continuous. So you see that this square in red on your correlation matrix is really strong A, but all the bins around are on the transition, so they have different color. And increase the number of compartments will not affect your correlation matrix, it will just affect the way you change the information from a heterochromatin to a chromatin. So you will see the transition on your correlation matrix with regions which are not completely sharp on one part will appear on your number of compartments. Performance requirement of test algorithm. Can you describe more, Wang, please?
I think because we should evaluate the performance for the algorithm. So we should test the time, the speed time, and we should test because I saw some paper. It's exist prediction. We should test the prediction or not.
For me, the performance and the complexity, I completely don't care. Some algorithms that I give you actually are completely slow.
Just the time.
The speed, because they want to compare every parameters during the review. But for me, the only thing that are important is, did you detect good TAD or not? Did you detect good compartment or not? If your algorithm takes 2 days on the cluster, you are a student, come on, you don't are here to make some things that are completely operational. You have to make some things that have sense and to understand the operation that you do. If you want to take 2 weeks on optimization, it's for your training, come on.
Okay. So the most important is just the speed, the time.
The speed, I completely will not judge you at all on your speed of your algorithm. I just want to judge you about, do you understand the features that you extract or not? Is the features that you extract are good or not? Okay. Maybe there is some other class where they can evaluate you on about your writing computer skill. You make some teach with BIP students. They are not completely good in algorithmics. Some of you have computational background and you love optimization. Some of you not. Here it's more about biology, bioinformatics. Understand your data.
Yeah, and try to make some critical assessments of what you have, and in a quantitative way. It's nice if you can have some curves, quantitative curves showing how the performance vary with respect to the parameters, but it's not so much about complexity issues.
Okay, I see.
Well, it's good if you can evaluate also that your code is more or less efficient, but the main goal is not to make it as efficient as possible.
Okay, I see.
Other question? What? I think I described everything from the slides. So for my initial plan, I said all that I have to say.
Okay, so maybe what we can do is to free you now if there